Morning and welcome to this presentation. My name is Georgios Imanidis, and I will be presenting you in the next 20 minutes or so a new technology we've been developing and we've developed and still working on it for the manufacture of orally dispersible tablets. You may know orally dispersible tablets are the tablets that you put in your mouth and they disintegrate and dissolve immediately, and so you don't need to drink a glass of water for that. And these tablets, or this delivery form, meets a number of consumer needs. For example, in terms of efficacy, they assure that there is a rapid onset of action. Remember, think of that headache you have and you want it to go away right away. And of course, in terms of convenience as well, you can take the tablet without needing a glass of water. You're sitting in this busy meeting and then you want this headache to go away, or you're traveling and so on. So in terms of convenience, they meet some customer needs. And of course, in terms of compliance, it's an easy administration. People who have difficulties to swallow big dosage forms, children, and of course, special need patients, mentally disabled, and so on. And finally, there is a number of therapeutic areas that this delivery form is relevant for. I mentioned headache already, gastroenterology, and so on. And as a result of that, there's been a relatively rapid growth, market growth over the last years. And it's forecasted that this growth will continue, this robust growth will continue in the next years for sure. The technology I'll be talking about is Lyopan, we say, or Lyopan, for the manufacturing of orally dispersible tablets. And there are tablets like this on the market today, and they are based on either one of these three technologies I saw you here. So they're manufactured either by direct compression or by freeze drying of a solution, or less important, using thin films. And I'll be showing you that the Lyopan technology has advantages over these three forms, and hopefully I'll convince you about that. So this is why. Let me start with the process, because many of the advantages are derived directly from the process. All right, we first form the blister. That's a conventional operation, conventional procedure. Form the blister and then dose the solid or dispense the solid directly into the blister cavity. All right? After the solid has been dispensed, we add liquid, again, into the blister cavity. All right? So we have a slurry, we have a wet mass, and this wet mass is homogenized by some mechanical agitation. Subsequently, it is frozen and then freeze-dried. The freeze-drying step is essential because that guarantees the immediate dissolution. You're familiar that, for sure, that for parenteral products, lyophilization guarantees an instantaneous dissolution of the powder once the liquid is added. And this is the same principle that we employ here. So freezing, freeze-drying, and then at the end, sealing. What does the solid powder look like? That's a generic formulation here. The API is part of the solid powder that we dose into the blister and can make up a lot of the whole formulation, 10 to 60%. So we have a high drug load in this form. We can have a number of carriers. One of two carriers, a binder, disintegrant, and very importantly for this dosage form, taste masking agent, and flavor. This is the solid part. Now the liquid may or may also contain a binder as well. Important is a wetting agent to make sure that the liquid really wets the solid the powder particles. And again, you may have taste masking agent, and of course the solvent, usually water. All right, now how much powder can we dose in these forms? That, of course, all depends on the volume of the cavity, right, and the bulk density of the powder. Okay. This is a typical form of a blister here. that we use. One can use larger or smaller blister cavities. And then as a function of the tap density, we can fill this and get at the end in a blister up to 1 gram of powder, maybe 900 milligrams. This is a blister that has, I know you cannot read this science from the back, it has a bottom diameter of 12 millimeters and a top diameter of 20 millimeters, so it's conically shaped. All right. Now, of course, you don't want to fill here completely to the top, but that's not a problem. You see here the depth of the height of the blister, that's a few millimeters, because once you add the liquid, there's some consolidation taking place. And so then here, if you fill it up completely, you end up with about 900 milligrams of weight of the tablet. But once you add the liquid, then the powder consolidates and it gets the height or the thickness of the form becomes 1 to a millimeter and a half shorter or into the cavity. We established nomograms to plan or to plan this the filling of the different forms, and of course, depending on the formulation, you have different tap densities and different forms of the blisters. All right, now back to the formulation principle. We have the solid particles. As I said, the solid particles consist of the filler and the disintegrant, and most importantly, the API. And then when you add the liquid, you add just enough liquid to fill in the voids. So the liquid here in between, what's marked blue, contains maybe a binder, a wetting agent, and a solvent. And this is what is then frozen. And after it's frozen, it's freeze-dried. This is a typical freeze-drying process. We freeze this in liquid nitrogen, and that's what you see here. So the blue curve is the product temperature. From 20 degrees, we go down to minus 60. That's the freezing of the liquid nitrogen. And at the same time, we monitor the electrical resistance. That's important so that we make sure that during the drying process, there is no thawing taking place. If there's a thawing, then the structure will collapse, and that will reverse the good solving properties of the attained by lyophilization. Okay. Then we bring everything back to a temperature of about minus 15 degrees. The red curve is the temperature of the cells, okay, or of the cooling elements, or the heating elements in the freeze dryer. After equilibration of about an hour, we draw the vacuum. This purple curve is the pressure in the chamber. And once the pressure goes down, drying starts to happen. And this is why the product temperature here decreases slightly. And from then on, we start increasing the temperature of the cells to accelerate drying. And at the same time, monitor the electrical resistance to make sure that there is no thawing taking place, as I told you before. And after a drying of about here an hour and a half or to 2 hours, we reach room temperature and ready to take out the product. Okay. In general, this drying time is relatively short. And I'm saying short compared to the lyophilization that is used for parenteral products. There, if you have vials, for example, you freeze dry overnight, 24 hours, or even longer, have really long lyophilization cycles or freeze drying cycles. This is not the case here. We have drying times of an hour and a half or two, depending, of course, on the size of the tablet. This drying can get longer. So, for example, for an 800 milligram tablet, it would dry for about 3 hours. For a 500 milligram tablet, it would dry for 2 hours. What you see here is the residual moisture content, and within this drying time, you can get a residual moisture of a couple of %, which tells us that the product is essentially dry. As a comparison, if you were freeze drying a solution, the way people do in general, it's done also for parenteral formulations, the drying times will be a lot longer. Look here, we have for a Lyopan formulation for a total weight of 130 milligrams. The product is dry, has a residual moisture of a couple of % already an hour after drying time. Whereas here we have 5 hours, and if we had a solution, a pure solution of comparable weight, there is still a lot of moisture remaining after 5 or 6 hours into the product. So the drying for a solution has to go on for a much longer time compared to the Lyopan formulations. And this is one essential advantage. I come back to this at the end when I summarize. It's an essential advantage of this technology that we can get process times that are short, or within a day we can run 2 batches, for example, with a drying time of about 3 hours. And that, of course, saves costs and so on. But I come back to this in a minute. Of course, product quality characteristics is important. First, foremost, the disintegration time. These tablets disintegrate within 3 to 15 seconds. The duration of the disintegration depends on the size of the tablet, obviously. If you have a big tablet, it takes a bit longer. However, this is all with the same formulation. So we run tests with tablet thicknesses here between 2.5 and 5 millimeters, and we get this disintegration times, 3 to 15 seconds, with the same formulation. If you do some of formulation optimization, add disintegrants and so on at the larger tablets, we can go below this 15-second margin. Then, mechanical stability of the product is very important. You need to be able to remove this out of the blister without falling apart. Okay, so it has to have a certain mechanical strength. We measure, among other things, indentation force. So we push a cylinder with a diameter of 2 millimeters into the surface of a tablet, and we do this to get numbers in order to calibrate the process. So we have 2 to 8 Newton of force in order to enter a depth of 0.2 millimeters, depending on what place within the tablet surface. And then when we push further down, then we break the tablet, and that's an axial breaking force. In tablets we measure the breaking force applied diametrically, so axially. We cannot do this with this tablet, so we have an axial force, and we get breaking forces of 5 to 40 newtons, depending again on the thickness of the tablet, and that's enough mechanical stability to be able to remove the tablet out of the blister and put it in your mouth without falling apart. We need to have mass uniformity, and I come back to this in a minute, but we can fulfill requirements of European Pharmacopoeia with respect to uniformity of solid dosage forms. There's some limits there, and of course, residual of removal, we need to be to a level of less than 1%. So when you get the tablet out of the blister, it should be less than 1% remaining there. So this is all specifications we're reaching. Coming back to the reproducibility of dispensing of the powder, all of you know that tablets, capsules, and so on, all the systems where we dispense a solid powder volumetrically usually, it's essential for these products to test the producibility of weight. The Pharmacopoeia demands that. I saw you data here of the filling. That's about 700 milligrams filling. These are 5 individual cavities in one blister form. And you can see yourself that the results are really reproducible. And the graph on the right shows you blisters with 5 cavities each, and this is averages of the weight of these 5 cavities, and we did this 6 times, and again we have really good reproducibility. You see the error bars here are rather small. We implemented this process for a number of APIs. This is just a few of them. The common candidates, of course, for this dosage form, painkillers, ibuprofen, paracetamol, and some other indications. And notably here, we can achieve the high dose required, for example, for ibuprofen, usually a 600 milligram dose, and it's possible to make a tablet, an orally dispersible tablet with a short disintegration time here with these APIs. So I wrap it up. We have some attributes of this process that are at the same time the advantages also of the process compared to other processes currently in use. I have subdivided this in product-specific and process-specific advantages. So product-specific, we can have a high drug load. We can administer drugs with a high dose and have a high drug load within the form. It's easy to work with poorly water-soluble drugs, but it's also no problem to work with well water-soluble drugs, so this is not a limitation at all. We have a really very fast disintegration time in the limit of less than 10 seconds. By the way, if you look at the pharmacopoeia, it defines orally dispersible tablets that disintegrate in less than 3 minutes. And of course, that's a joke. Nobody will put a tablet in his mouth and wait for 3 minutes. So that we're here a lot better than what the official requirement is. And we're aiming at less than 10 seconds disintegration time. Of course, we have the possibility for taste masking. Taste masking is very essential for this dosage form because if something is, taste past in the mouth, people then will not comply. And there are APIs, think at ibuprofen, that are extremely bitter, and there are taste masked variations or qualities, grades of these APIs on the market. They're usually manufactured by coating the particles of the API so that they cover their taste. And it's possible to use these already taste-masked APIs in this process to manufacture orally dispersible tablets because we dose the solid ingredient. And of course, we have a better mouthfeel compared to the direct compression. That's the effect that comes from the lyophilization that gives this, this immediate dissolution. Now, process-specific, we have short process times. That's quite relevant, not only because lyophilization is an energetically expensive process, but it has to do also with tying up of equipment. You cannot afford to lyophilize for 12, 24, or 48 hours for this kind of product. This is way too expensive, and we improve significantly on that. Of course, we have high production efficiency, and at the end, there's energy and cost saving with that technology. So, as a summary, again, we have a considerable customer need, and it's growing. It will be growing. It's forecast to be growing in the next several years. It's an attractive market potential, and this is not limited to OTC. We can have prescription drugs also administered this way. Think of the special needs patients, mentally disabled, and so on. We may have some prescription medication administered there, and then of course we have a number of advantages of this Lyopan technology for the manufacture of ODT products compared to whatever is on the market at this time. And at the end, I'd like to acknowledge, of course, my colleagues. This is a typical, I say, academic presentation. I'm here on behalf of the Pharmapan company, but myself, I'm a university professor. We work with them. And of course, I could not finish without thanking the people in the lab who did the work. So Yael Gali and Michael Lanz, working at the School of Life Sciences of the University in Northwestern Switzerland. Thank you for your attention. I'll be happy to take your questions. Yes. Okay. I repeat the question so that probably you haven't heard it. The question was shelf life of these products. The shelf life for these products will be influenced heavily by the residual moisture, very clearly. So we have to go down with that as much as possible. And then the second will be packaging. So the material of the blister, and we use, I must admit, a lot of aluminum for these blisters here. So the material of the blister will affect shelf life significantly. Yes? It's an alu blister. Copper foil is the alu as well. You can have an alu blister or an alu PVC blister, depending on the situation. Yeah. And I must say, these are peel-off blisters, so you don't have to push the tablet out of the blister. You peel off the top layer. Yes.
Professor Georgios, very nice talk. So when we compare Lyopan technology with the Zeiss technology, we have some difference?
Okay, the question I guess I repeat the question. If we compare Lyopan with CyDis, yes, the difference, we believe there is a difference. The difference consists in how the product is prepared. So we dose a solid material and the liquid on top of that. As far as I'm familiar with the CyDis technologies, there is only one dosing step of a solution or a suspension with a certain concentration, of course. And I think, or we believe that with the Lyopan technology, we have a bigger flexibility in varying the drug load. There is only so much one can dissolve or suspend in the liquid that is dosed. Here, there is no limit. We can dose the, the solid form. So we have a higher flexibility in this respect. Plus, then, we can limit the number of liquid we add. I mean, we add just enough liquid to fill the voids within the powder, and that shortens the freeze-drying time quite a bit. So these are the two major differences. Plus then other things I mentioned, taste masking and so on. I think there is a bit more flexibility here compared to the CyDis technology. Again, as far as I know, the CyDis technology, I don't know if there's a Catalan person in the audience, but I think, yes, we have advantages in these points, yeah. Yes?
Have you scaled this up, or is this just a laboratory?
Okay, the question was if this is scaled up already. So all the results I saw you, we received these results with a prototype. So the company has manufactured a prototype and placed the prototype in our laboratories at the university, and we ran this test. The lyophilization we've been doing so far in a commercial, in a normal commercial lyophilization that's used normally for parenteral products. And this is the current state. At the same time, I mean, we do formulation development, process optimization with what we have, and at the same time, we're looking at scaling up our setting up a commercial line, which, however, does not exist yet. Okay. If you, for more commercial or business information, I'd like you to refer you to the website. Here, there's more information there on the business part of the application. I'm sorry? The presentation, yes, well, as far as I know, all the presentations are with the organizer. I don't know what the organizer will do with the presentations. Again, I asked before and I didn't get a clear answer. But if you want me to send the presentation directly to you, I'll be happy to do it if you give me your business card. Okay. In this case, we close this. Thank you for your interest and the discussion, and see you around.